Eventually will come.
Is it the pomponi?
We haven't seen this one. It's right here.
So we just gonna get some images of this guy and confirm its species. So compared to the black swallower, however, the black swallower does open its jaws even larger than the gulper eel. The black swallower, Chiasmodon niger, a fish that bites off more than it can chew, and it often succeeds. Famous for swallowing prey much larger than itself, this fish can consume up to twice its own length and many times its own body weight. We do have some interesting imagery of the black swallow we saw recently. It's been going viral, so you can check out SOI's channel to get a look at that interesting and amazing imagery of the black swallow.
Yeah, let's take our time to get good images with and without lasers. Really good details, and then after that we will take it from the base as we did with the last coral in here.
Okay. Lasers on it.
So with the lasers on, we want to try to get some imagery here. Remember, all lasers are set to 10 centimeters apart, so we're able to tell the actual length of this specimen here. So by looking at its morphology, we're able to determine what family it belongs to and possibly its genus.
So after we do our shots, we will give space for them to film, and then
Sure. So we'll just take a few more images.
Time to get one really good for us.
Possible carnivorous sponge with associates on it. Really amazing imagery. So you'll probably see us disappearing for a little bit as we work on one of our BBC projects.
Look at all the scale worms.
Yeah, scale worms. So we definitely have some scale worm associates on this almost extraterrestrial looking sponge. I put it to be about 25 to 30 centimeters long. Our sponge experts have just come into the room and are quite excited. We have with us Dr. Shirley Pomponi, Dr. Megan Conklin, and our very own sponge Rob, Mr. Rob Cassidy, who's working on eDNA associated with sponges. And they're all in the room, bright-eyed and bushy-tailed.
Do a Niskin. A Niskin.
So we buy a Niskin. So we're just going to fire off a Niskin bottle with samples, some water close to this sponge. Rob is doing work on the eDNA that he's going to extract from this water sample. So we'll be firing off Niskin bottle number 3. So that's Niskin number 3 being fired. We'll give it its own number, 472.
light fell off.
I think the light fell off.
We'll get that out. We'll get that after. Just put the handle in there.
We'll get it later.
It's not my light to run. How did that fall?
All right, so as you can see, we are collecting the sponge. For right now, we're just putting the light away. We're going to have to go pick that up in a second. But yeah, we'll pan back over to the sponge here in a moment.
So we're live now.
We are live
Okay,
now.
great. Hi, everyone. It's Shirley Pomponi, and I'm in sponge heaven right now. I'm a sponge biologist, and about, I guess, 45 minutes or an hour ago, we came across a carnivorous sponge. It's called a ping pong ball sponge, or if you want to be more dramatic, the death ball sponge. Its scientific name of the genus is Chondrocladia, and you'll see you'll be able to see it in just a few seconds. We've got it
Still there.
in the arm,
Porch up super gently, okay?
and we've been watching this sponge for the last probably 3/4 of an hour, getting some really good imagery. So it's got this central stalk from which project all these branches that have balls on the end, and the only time I've ever seen this before, I've not seen these little tiny balls at the end of those branches. And what we're thinking, in fact, if you look at the one kind of on the bottom, it almost looks like a circus balloon that you start to blow up and it's getting inflated at the base. And that, what we're thinking is the formation of the larger balls. We're not sure if that's how they're capturing their prey. I'm going to need to consult with an expert, Jean Vasselet. In fact, I was just getting ready to email him and ask him. So those of you who are on the chat, please don't do that in advance. Yeah, but we're seeing all like really interesting structures that look like they're the formation of the larger balls that are very characteristic of this sponge. So the one that you're seeing, the very smaller one, looks like it has some structures, maybe some spicules that it uses to trap the prey. And we're also seeing some pigmentation in the stalks. Maybe if you pan up a little bit higher. There you go, where it looks like it's kind of pinkish, and that may be from what it had. The sponge is just digested from a shrimp, for example. So we won't know until we have a chance to look at it more closely. But yeah, you can see some of the ball structures have kind of like areas that are pinched off. So it may well be that the sponge captures the prey while the ball structures are very tiny, and then the ball encapsulates them, and that's where they're digested. In fact, Robert Cassidy had just asked about that a little while ago, so we'll hopefully be able to confirm that. So carnivorous sponges were first discovered in 1995 by Jean Vasselet and Nicole Bourriasneau in France, and they found a very small, tiny sponge in this cave that they explored off the coast of France near Marseille. And when they took it back to the lab and examined it, they realized that it didn't have the usual structures that most demo sponges have. This is not a glass sponge, this is a demo sponge. It didn't have those structures, the little chambers that set up the water column going through the sponge that enable it to filter particles out of the water. And when they examined it more closely, they could see that the sponge had on its surface these microscopic skeletal elements called spicules that are made out of silica. And these spicules that are on the surface of the sponge, we can't see them here because they're really microscopic, are shaped like hooks. So it creates what's like a Velcro surface on the sponge. And when some unsuspecting tiny shrimp lands on the sponge to start picking particles off of the sponge surface, it gets trapped in the Velcro spicules, and it can't get away. It's stuck. And what the sponge does then is mobilizes cells that completely engulf the prey, the shrimp, And then these cells start digesting the prey using same kinds of enzymes that we use to digest our food. And then the only thing that they can't digest, to the best of my knowledge, was the chitinous exoskeleton, the shell, if you will, of the shrimp. So they get kind of cast away, spit off. But then the cells of the sponge then transport those nutrients throughout the sponge. So we're really excited to see if we can get more information about this process of capture and digestion and transporting of the nutrients with this sponge. I don't know if this has ever been recorded before. I can tell you that I've never seen anything like this before. I've never seen these really unusual pointy ends of the branches. The only time I've ever seen a chondrocladia before, and even in pictures, but I'll have to check on this, they're all inflated spheres. Anytime you're ready.
Let's see how the collection goes.
So there's a scale worm that's living on this sponge as well, and it may be a symbiotic or mutualistic association. We're not sure, so we're going to try to get the scale worm as well.
Porch out and then see where you're at. Okay? So what you got, so we're at.
Seriously pointing out that it's actually eating this shrimp that's caught on it right now.
Which one?
Maybe around
Yeah,
the
that one. Okay. You want to zoom in on that again?
Yeah, if you want.
Can we just zoom in?
It's on the
Was it
top.
at the top in that area where it looks like there's an onion ring? Was that the area?
Yeah, right there.
Right
Looks like an amphibia.
there, right there. So, zoom in on that. Oh, you think that's trapped there?
Yeah, that's what this is.
Oh, so let's see if we can zoom in on that. This one.
Yeah, I was trying to see if
There,
it
yeah, I
walked
know.
around earlier or not.
Yeah.
Difficult to say.
It-
We've seen
It
amphipods
looks
on
like
the,
it might be an amphipod that may be trapped because it's not moving. Very cool. That's very cool. That's great.
Another little-
Thank you guys very much. That's
little
great.
amphipod or another little worm. I don't know. Anyway, let's see how the
Could the
collection
scale
goes.
worm be stuck? I think they're probably okay. Easy mountain. Look. You got room. Alright, coming out real slow. This guy's going in the big one, right?
Yes. Yeah. If we can do. How lucky am I that my dive lead hot seat watch just happened to be now?
Backing up. Alright, he's gonna go in here. I'm just gonna go super, super
It's
slow.
still not
And then
exactly
when
there.
it comes time to put him in the box,
No, no, hold on.
when you're about to let him drop, when you drop it, you get the arm out of the way of the lid, because as soon as it hits the bottom, that guy might try and skedaddle, so I'm gonna shut the lid real fast, okay? Alright. Whenever you're ready. Go for it. Kind of rotate it real slow to where he's facing down too, if you want. Help start him in the box, you know. top there, see? Shoulder left a little. Shoulder left a little bit. There you go. Sorry. Keep rotating.
Keep upside down.
Yep.
So since 1995, when carnivorous sponges were first described by Jean Vasselet and Nicole Boreasneau, there have been 130 new species of carnivorous sponges that have been discovered and described.
Yeah, I see. Like,
Okay. And you can just kind of tap this joint down.
Sorry. So this is, Juliana, is this 473?
This is 473.
Thank you.
Straight down from there. That's it. Tighten the lid. On the lid again. Shoulder left a little bit. There, see? There you go. Straight down. Don't come right anymore. Turn the lid again. Let's stay to the left. There you go. Straight down. Don't come right anymore. To the left. You can tilt this joint to the right if you want to get it more that way. With your shoulder up. There you go. Alright. Straight down. That's perfect. Now just coming down. out of there. Yep. Just get out of there, but don't hit nothing. No close. Go for it. Nice.
Perfect.
And we got a worm.
Excellent.
Got everything.
Excellent. Thank you.
Yeah,
Wow.
You might find a trick to pick up this light because I think it's snapped
Yeah, I think
at
it's
the
round.
handle base.
I think it's perfectly.
I get the scoop situation in.
We'll see. We'll give her a go.
We'd like to challenge you.
We'll give the old college crap out try again.
All right, we got to go
Ultimately,
pick
it's
up a
not
light.
mine, so.
Oh, so you're saying leave it?
So not
Roger,
that
leave it.
bothered. Probably the only bit of kick it is.
Yeah, so today we were using a different light. So past couple dives you've seen we had a ROV light modified to be able to be carried in the manipulator. But today we had a light with no connection to the sub, but a little handle. But it, of course, came off the handle, and it's laying on the seafloor now. So we're just going to have to pick it up. Should be good.
Thanks, Alex.
Alright, let's go look at this thing. I'm gonna go over there. It's too long. Yeah. Yeah, it's just the handle just came off. So.
It's been on since 11:30 last night. Quite impressive, not
Oh, not bad, right?
Oh, the battery.
Yeah.
Battery's lasted.
So this
Might
dive
be able to
started,
have a bit of clips, actually.
oh gosh,
Yeah,
how many
as long as
hours
it's not
ago?
dangerous, open
Probably
or
about,
something.
Now, like at this point,
Almost 10 hours.
yeah, almost 10 hours ago. We're on this feature at almost 1,100 meters deep. We're on this feature that it looks like a collapsed caldera-like mound that was mapped during this
So,
expedition.
I think Johnny's right. I think we could
And-
probably either get it by that or it looks like there might be a very thin rim.
yeah, there is a rim there.
But I don't know, it might just pop out of there too. Where do you want us to get it,
Ooh.
Johnny?
Wherever works. I mean, there's a theory there's a lock on the flip, so without pressing another button.
Okay, well, we'll
It's
test
always
it.
a theory.
Exactly.
We will test it then.
As I say, it's not mine, whatever happens.
So our plan was for this dive is to continue moving along the rim of this kind of caldera-shaped mound. Oh my gosh!
I mean,
I would want
I'm going to shut up
if
while
anything,
they're
the pressure
while
will
they're
keep it shut, so.
trying to maneuver to pick this up.
really flush, so yeah.
Yeah, right there. Maybe move up a little so that it gets even between your tines, you know what I mean? It's gotta be, I think you gotta grab both sides or it's just gonna slip out. It's gonna be tricky. Get me to come around to the side of it where you can see it on the side a little easier. So, yeah, we guess we could try the net though.
Yeah, we could.
I think he's gonna get it. Let's see. I'll try it. Yeah. Nah, slipped right past it.
How wide does the arm go? Can you grab it?
I
We
don't
have-
think so. We could try, but it's,
We have a few tools in our toolbox to try here.
it's a wee bit too big.
More
If we
than
can
more than a
see
wee
it.
bit.
I think he
Yeah.
just didn't get it because we can't see his all the way.
Camera's not in a terrible position on the net. It gets a little worse when you turn forward.
both fingers touch it, it'll be okay. That's gonna be the trick though, making sure it does. Now rotate it. Now rotate it a little bit clockwise. A little more clockwise. Keep going, keep going, keep going, keep going. Keep going, keep going, keep going. Alright. Alright, Johnny, there you go. For help, contact. Those people right there, okay? You got that? Go ahead and put it in.
Someone on the chat said, I know I borrowed your light, but in my defense, it wasn't well connected. Oh, God.
Probably it's right in the back of the chart over there. You don't have to worry about using that two-handle again, so.
While we're here, is there any merit in giving a little Charlie a whirl?
I mean, that'll be your call, but it's pretty,
Is it just sand, is it?
it's pretty dull.
Yeah.
You know, I'd look for more boulders or something probably personally.
Alright.
Yeah, we can find some sort of crevice or fissure, but there's a danger of it falling into.
Yeah.
Boy,
We could do risky shots.
that's right, that's right.
That could possibly go wrong. Does little Charlie get it?
No, BBC.
Well, there you go.
Exactly.
Decided into an active volcano.
All right.
Okay, now what?
All right. Now we want to continue on. What are we at? We're at wait. Yep. Okay. We're going to continue on towards waypoint 11. Excellent job, guys. Excellent job. Thank you.
Nice arm in there. Yeah, it's quite a stop there. Huh?
Yeah, quite a stop.
Yeah, worth it.
All right, come on up.
So it should be this those guys make another one at the bottom of that blue one you've got.
Oh, and you could put your GoPro or whatever
Yeah.
now. So Alex was showing me too. You can 3D print them out for 'em.
What are we 3D print?
For the blue.
That's the
Oh.
company that makes that light.
Yeah, yeah,
So that's a camera though, right?
Yeah, that'll be a camera.
yeah.
But I
There's,
think we.
There's a mount for GoPros, so if that takes like, if you could do like the GoPro adapter,
Yeah.
whatever the heck they are, the knuckles. They don't sell it anymore, but they just have a 3D print for it. No, but if that has a if it has the knuckles for the GoPro system, this is just a mount that they have, and it fastens onto the bottom 3 screws of the battery housing. So you can wedge it on there. But yeah, people strap all sorts of things.
This company that made the lights on the GoPros, I think it's called Angle, everything's sliding.
Okay.
They're probably going to come there.
Yeah, that's
Making nothing really.
I think we actually figured out the 4K video on that thing just in time for you to not use it during.
yeah, of course. Yeah, I've been on this site before for looking at little Charlie.
Es la mejor manera también
This thing here? Yeah, that's the blue ROV that we've got in the office.
Nada compartiendo los videos. Can we zoom on this
The
coral?
corals?
What do we?
Can we zoom in this coral?
Yep. Can you zoom in on that coral, please? I'm still giddy over the chondrocladia, over the carnivorous sponge. Oh, lots here on this rock. We've got the
What type of coral is
ctenophores. We've got, I don't know, what is that? Stylaster. Some more sponges.
Maybe we can stop. Do you think it's alive? Or it's like
Which one? The
yellow one.
do I think it's alive? Yes.
Yeah, I mean, it's alive, but we've seen like some that at least were at the end were so antics. But I think this was.
Oh, right. That's right. We did get that before. Let's take a look.
On the rock.
Divas have to collect.
Oh, I see what you're saying, Bea.
Coral and a few.
Yeah, we saw something a few days ago that was actually zoanthids encrusting on a
Yeah,
dead
but I think,
stalk. But
I think this is.
yeah, when we get a closer look at that, we let's see. Yeah.
I mean, that's a massive.
What do you think, zoanthids? You think zoanthids, Juliana?
No, I think
You
it's,
think
it's a coral.
Octocoral?
Yeah.
Let's
We
see, let
should
me
collect
count.
this one after we
Yeah.
get the images.
So if you see them,
Yeah,
and that's what I said. We don't have space for the whole thing.
Oh no, no problem. This is okay.
do it for the lantern.
A piece.
Okay, you don't want the echinoderm, right?
I think.
No.
No.
Okay, all right, so what we'd like to do is snip a branch off of that, please.
Got a cover.
We don't need the, we do not want to collect the echinoderm.
Where's that gonna go? Quiver?
Well, depends on how big it is. But
Hey, do we put something in two or no?
We see in 3D
there
are pretty open.
is,
Two's open.
Oh,
for the quivers, yeah, two.
Two's open,
Yeah.
All right, so.
However, wait, did something go in one? A purple coral?
Yes.
Yeah,
Yeah, we did
purple
put something
coral.
in one.
Yeah,
Oh.
but let's get some good imagery because I think we might collect also the ophiuroid.
Oh, you want to collect the ophiuroid too?
Sure.
But this way, let's take some with images and zooms while we figure it out.
Cheryl,
Yes.
if you don't have it there, in one it's 467. If they didn't write it down.
Yeah,
Quiver
it's here.
one.
It's here.
It's here? Oh.
Yeah, it's here. Thank you.
They're still beside, but yeah, it sounds like a roof.
And who's behind it?
Stand on.
There? Who's this
What do you
guy?
think, Bea? Ophiuroid? No ophiuroid?
I'm asking you.
Okay.
If we do get it,
It's going to be hard to
well, I think it'd probably be easier to just clip the whole thing at the base.
Yeah.
write down. No.
Yeah, and then just get the whole thing together, cause he's not gonna come off of it.
No.
Oh no.
Yeah, maybe.
So we could probably fit all that in the bio box.
Yeah.
One of
One of these. Yeah.
Oh, look at that. Would you look at that?
How big it is?
How deep?
How big? It will fit on one of these, right?
I think so.
How big is it?
Oh,
So,
okay.
but if we do that, if we need the ophiuroid, we're going to have to collect the whole
Oh
thing.
yeah.
Would you like
Is that what you want to do?
Oh, it's not that big. Yeah, it's like. I think we've seen a bunch of these, but we haven't collected
Okay.
the yellow ones.
the
Okay.
whole thing?
Okay, so we want to collect as much of it as possible. So maybe kind of, if you can snip it down there, that would be good. We've seen a lot of these. We haven't collected any to study them further, so we're going to try and do that and be able to get both the octocoral and the ophiuroid.
So you want the whole thing, and we're gonna put
Yes.
it in,
Maybe two,
I don't know what
2A or B,
two what?
either A or B.
Or 2A or B.
Yeah,
Red.
what do you think? Unless you think it'll fit in one of these.
If you like it, I love it.
Which one do you want? You think it'll fit in one of these smaller ones? No, pushing it.
Probably be
All right.
I think that we will
bit tight.
Bit tight. All right. Is this like for me?
No, you want
For
to
both of us.
Well,
You're not good at sharing?
Come, we can share.
I didn't know if you wanted me to open it or not.
That's big enough for the whole class, right?
Yeah.
No, it's como de sacar en uno de los bioboxes.
Mm-mm.
Es como un
Es el que
tipo-
parece.
tipo mushroom. The red one.
This one, this guy.
After you pick it up, if we could get some close-up video, please. There's a rooster in there.
Got it. It's that dark chocolate stuff, huh? Medio amargo. Oh, that's all.
All right, good. Good.
Well, bring him up for a glamour shot.
Yep.
Beautiful.
Sure.
And if you like, yeah, tilt it a little.
Oh, it's beautiful. All the colors are popping. This guy.
He's cool. Of course you.
It's real nice oak.
Wow.
Wow.
Marco.
Oh, marker.
Always.
Okay.
Okay.
You happy?
Yeah, we're happy.
Alright, we'll porch out nice and easy.
We're gonna.
Four,
it's
seven,
gonna
four.
go.
Alright.
I'll let you know.
And that'll be 474.
Yeah.
We're gonna go into
Es un octocoral.
Es una temporal. Puede ponerlo como comentarios.
Do you want to give it two numbers now?
Sorry?
Do you want to give us two numbers now? For this,
The number?
You want a 474 and 475 for the optical and opioid? You want to do that now?
Oh, I'll put it as associated.
Or you want to wait and do
Maybe
parent?
later.
Later,
Yeah,
will you
to
do
avoid-
parent? Okay,
avoid
got it.
mistakes.
All right.
Yeah, I put it as associated.
This is number
474.
starting there.
474.
Mm-hmm. Alex, you gonna answer that?
Yeah.
Okay, thank you.
Yeah, so bringing samples up, we can see some issues with pressure changes, but a lot of the really traumatic barotrauma, right, trauma from changes in pressure, a lot of the dramatic stuff has to do with fish that have swim bladders, which are gas-filled, and so that gas really expands. But for things that don't have pockets of gas in them, it's not as much of an issue. So like this coral, for example, isn't going to come up all expanded and bloated. But if we were collecting a fish that had a swim bladder, then yes, bringing it up would cause
Thank you.
barotrauma. So on most of our samples, not really a consideration.
Yeah, you might mention, Alex, that most of the effects that we see are from temperature, not.
Oh, a
pressure.
lot of them,
Okay, yeah,
a lot of the
those
effects that we
pressure
see on our samples are actually going to be from temperature. Shirley wanted to note, which is something we've seen before. For anyone that remembers the dives where we were collecting the Riftia tube worms and bringing them up and keeping them alive to study their bacteria, we ended up having to use like a giant camper cooler as the bio box because that would insulate them from the warm waters near the surface that was killing them. And so in order to get them up through that layer of warm water, we actually were putting them inside of a camper cooler on the ROV at depth, bringing that up through the warm water, and that was insulating them. So yeah, the temperature is much more of a challenge on some of these samples.
Okay, well. Rachel, are you up next?
Yep.
Oh, okay, cool. All right, well, my hour surely has gone by fast, and I'll be back tomorrow, I think, in the hot seat, taking part in the dive. We're really fortunate because we can actually view the dives just like you can on monitors all over the ship. So in our cabins, in the mess hall, and the science labs, we can see what's going on. So it gives us an opportunity to kind of pop into mission control if we want to look at something more closely. So really great facilities on board the Falcor too, to be able to, for even us here on the ship, if we're not in mission control, to participate. Okay, enjoy the rest of the dive, and I'll see you tomorrow. And Dr. Rachel Lauer, our resident geophysicist, will be on for the next hour, and you're in for a real treat. Okay, so we're heading, thank you very much. That was excellent.
Thanks,
And we're just heading towards Heathrow. Okay.
Yep.
Yeah, so we collected two samples.
What's up, Juliana?
And we're just headed towards point 11.
Oh, yeah, 11. Yeah. Yep, the pinnacle. It's the summit. The end of the day. It has been all, it started that way.
I know.
We saw a cat shark.
All right. Thank you, Zach.
Thank you. That was a great class. Thanks. I learned a lot within an hour. More
Me
about
too.
sponges in an hour than
Me
in
too.
all in the ground.
I sent the files.
Okay.
Those are like,
Hello. Hello. Good morning. What?
Great, Jarvie.
Oh,
Go ahead.
I'm not really awake.
Can you start moving the vessel?
Yeah.
Right now.
Driving while sleeping. It's another
okay,
kind
we're
of
in the water.
hazard. It's not illegal.
I'm going up 12, I think, right? So probably just somewhere like, that. We're going 12 now, right?
Yep,
Through 11.
through 11 up to the summit.
Wow, we're almost at the end.
I know. I'm going to start and end this dive. Yep.
Must have good stuff in the middle.
No, I saw some of it. It started strong, actually.
We'll still be on track at 2.
I think so, yeah. We are gonna, since we are running a bit ahead of, well, we have some time, we're gonna do a heat flow if we can. Because it looks soft, which I did not expect.
Yeah, me neither.
Did not realize there was so much on my glasses. Oh, what was that?
What? Fish.
Nothing. The whole world is responding to a very strange raccoon in Seattle called Jimothy. Yeah, and now I'm seeing Jimothy everywhere, even in the deep ocean. He's, yeah, he has like some kind of spinal deformity, so.
Like a raccoon if you just get the cherry tomato.
Yes, exactly. He's insanely cute. Yes, like a round. Did you see him as a baby? Oh my god. Unbelievably cute. What? Are you sleeping? Free association back there? Yeah. So we're going to end this dive with a heat flow measurement. We started it that way. I think it was like 10 hours ago. When did we start? What time is it? 10. Yep, 9 hours ago. Cool. Let me text Roberto. Cool. Going up the steep slope. Cool. So today's dive has been on this kind of crazy looking structure that's like a kind of like a caldera volcano with like a ridge around a couple of smaller what appear to be mud volcanoes, and we spent most of the time so far on the ridge, and we started climbing up onto the ridge from the sediments, and it sounds like it's been an epic dive. I took a map in the middle of this and just checking up on all of the texts. We found lots of cool things, from corals to sponges, lots of evidence of methane. And we're going to finish on what is the summit of this structure. We're heading up to it now. Oh, nice little sea star.
not keeping up with me. That's all right. I'm used to it. So this is the high point of this that we're going to, right?
Yep. You know, just the
Say we're
king of the mountain.
Put a
Yeah.
flag.
Say we're there, put a flag in.
Later, some of these rocks to see what it's colored.
Yeah. You want to zoom in on the?
The red,
Yeah,
the red
sure.
ones or
The big ones.
Big ones?
They are like covered.
In anemone or what are
oh,
those?
these guys?
Polyps.
They're
Yeah. What are they?
Zoanthids.
Zoanthids.
If one of them is loose and tiny, we can put it in a bio box. Yeah,
There's one of those is like a shell with lots of them on it. Or a
You want this one right in front of us?
Yes, please.
yeah. Cool.
Well, what about this one? Might be better. These are a little smaller. Easier to grab. That guy?
On the
Yeah, I thought that would be better.
the little one. Yeah.
Yeah.
Or below. Yeah, whichever.
Sorry, it's cloudy.
Oh,
You think that one is too big?
this
I mean,
one.
we'll put the arms out and we'll see, but it kind of looks like it might be.
It's like this one has these ones, but a bit bigger, so
Yeah.
that'd be better for ID.
You mean this one? Yeah. See how? What room do we have? Buy a box 2.
And you also get a rock.
And a rock. Bonus rock.
If I get a bonus points, right?
Bonus.
From here.
Are we getting which one out of rock?
We want this one bits and the rock. So maybe we can let this.
So we're going to select one of these rocks with zoanthids and other tiny communities clustered on it as a
You
sample.
can see if we can get it in the
Got-
jaws or not.
It's trying to pick the right one.
Just careful not to hit the blade on it, 'cause it would grab it with the blade.
Now, some of these rocks have been very brittle, but these, not sure. Yeah, even that shell has like tons of them on it. Get a measurement at the pinnacle, if there's enough sun.
Probably gonna have to go.
I mean, there's a segment we can
clockwise.
always
Yeah.
go back.
We can go back to the where we
that
were.
escarpment
Yeah.
on the left.
Yep.
I mean, we have,
We have time, right?
but
What is the scheduled ascent time again?
we were supposed to come up at 1.
Oh, wow. Okay.
So when I arrived, we were already at waypoint 10. I think we didn't explore the waypoint.
¿Tienes que lo pongo como un sample biológico entonces?
No tenemos,
¿Qué no tenemos?
no tenemos sample biológico.
Yo no sé.
Ah, what you say, what you say.
Sample biológico.
Los sedimentos.
No, no.
So are we aiming for Biobox 2 or?
You tell me, boss.
I think it looks too big to go in the. I can't tell. How big is it?
Pretty chunky.
Yeah, might as well put it
We're
in.
coming up anyway, why not do 2B, right?
Yep. 2B or not 2B.
We used to have that on the list.
Did you? I just
They
had
made-
to say
they
it.
made me
Sorry.
take it off.
Oh, God. They made you not have your dad joke on there?
It
Oh,
was on there for like a dive or two.
okay.
They didn't make me take it off because somebody kept
Where
taking
is
it
it
off.
going?
Oh. Oh, you let them have it. Yeah.
Very serious.
BB 2B. Biobox 2B.
Is it going to BB2B?
BB 2B. Number 475.
I would estimate that maybe we need at least 40 centimeters of it. Looks all there.
Yeah, that is a big rock. See, somebody appreciates her sense of humor.
So I think that
Uh,
It's very red.
yeah.
What would you expect this to be, like a sandstone or?
So all of these are primarily some form of a carbonate.
Wow, that would be good.
Yeah, and then if you have iron oxide or other components that make it red,
It's right this way. Yep. So, like a limestone is a carbonate, is it?
yep, exactly. Yeah, so all of the seep sites are forming carbonates through microbes and chemistry and and basically all the byproducts of their metabolism. So,
Are you a Star Wars fan?
of course, I am.
Isn't that what they put Han Solo in?
No, that was oh God,
Carbonite.
carbonite.
Very closely.
It was a close relative of carbonate, but black. Yeah,
And more almonds.
and much more ominous.
No.
Although actually a lot of the carbonates here are very dark, so maybe they deserve a rebrand, like carbonite. What was that little fish off to the left? Yeah. Oh,
Like a fishy fish. They're different colors too. Aren't they?
yeah. Yeah, we've seen everything from like red, black,
It's like green a minute ago.
greenish, yeah, just depending on the conditions, water conditions, and under which it formed, whether they were oxygen rich, poor. I assume that those are due to some kind of iron.
So that's what they're everywhere now.
They are. ready to head to the summit?
Yeah.
Should we explore more?
Those rocks seem like reddish, right?
Yeah, I think that's probably like iron oxide,
Yeah.
kind of. Still carbonates, but they
But
can
not
take on
cement.
whatever color. Yeah.
Like the tubes from yesterday. They
Exactly.
were like reddish.
Yeah.
Reddish.
Sure. Rage RV, resume the move. Oh, I changed it. Yeah, see, you can push a little more than us. But if we're recovering anyway, it doesn't really matter.
Oh wow, the zoanthids are covering everything. Wow. Okay. Maybe no heat flow up here, but we will get a look at something cool. Wah, wah. In the interest of having it see another day, we might not.
We've got another.
We can
We got 2 more. We can do it. Just kidding.
The porch won't quite come in. I might have to push that on sometime.
Tired.
Yeah, it's tired.
I get it.
Like me, old.
Old and tired. We're not old.
So
Just
you, that's what I mean.
seasoned. No, I think we're probably the same age, so I'm speaking for both of us.
You're old in spirit, Julian.
I know. Turn 30 and it's just.
But you feel like
Decaying.
50, 60.
Straight down here. All right, I'm gonna try and push on this a little bit.
Cool.
Oh,
Oh, does that work? You can like just give it a nudge. Trying to nudge the porch in. A little help from the rock. Oh, it worked.
it's like the old tractor sometimes.
Yeah, you
Kick
know.
it on the back left tire and-
I've got a 1998 Toyota Corolla. Needs a little kick sometimes. Hedgehogs, somebody said. That's awesome. It does look like hedgehogs.
Looks like Timothy.
Jimothy! Oh my god. So glad he's come back into the chat. Oh. Jimothy is what we all need. What's that?
Not even.
I know. I can't. I'm really sorry. I'm trying not to talk about him,
Am I going to the waypoint or the top of this?
but what?
They don't look like they're the same.
No, I think the top. Yeah, I feel like we should finish the dive at a pinnacle. This is the highest point. But we'll explore to get some heat flow, obviously. Yeah.
I was gonna say we'd be recovered at 12 if we leave now.
That would be too soon. We can't do that. Look at that. Look at all these zoanthids.
Okay. Explorer mode.
Fuzzy.
Yeah, what is that?
What kind of shells are these?
It doesn't something alive?
No. No.
They don't look like our normal mussels.
Oh, they're clowns.
So one of the things we do on these cruises, if we can find styrofoam, since it's been primarily banned in most places, is drop and color them and then sink them into the deep ocean. They shrink down into this hard, cute little shot glass sized cup. And for my cup this year, I chose to draw Jimothy.
Oh no.
Yes, I did.
Is he on the vehicle right now?
He is, no, because I haven't finished him. But, I mean, it was 1 a.m., so decisions were made.
You're
And
up at 1 a.m. drawing Timothy on top?
Jimothy was drawn at, no, I guess I started him at midnight. But, wow. Is this like this is a lot of zoanthids, right?
Yeah.
Like a strange number.
Like famous it's full of these.
Oh, okay.
Bunch of rocks kind of over this way. You
Oh,
can go see him?
octopus. Can we-
Octopus.
Just
Right.
you pass it. Just
Wait.
we need-
He's back.
I was looking at the sonar.
Right there.
There he is, right there. Right, this little, right there. A wee one.
Yeah, he
I
didn't
know,
spot
he's
you.
very camouflaged.
I'm gonna come around.
We have a wee octopus.
Creo que es el de ayer, ¿no?
Yeah, they don't.
Tater. What you mean tater?
It's like different annotations platforms.
They
Oh, I thought that was his
Imagine.
name.
I don't know.
We can name
No,
it
we
Tater.
can name him Tater. We could name him something else.
It's a good octopus name, I guess.
Maybe not naming
I guess y'all probably want to zoom with this guy, huh?
Yeah. Oh yeah.
Oh no.
Do you know?
I didn't.
Were we only collecting them now?
Not the species.
Hmm. Wow.
Pequeñito, pequeñito.
Hmm.
It's a octopus.
Musa?
Yeah, moose octopus.
¿No es el mismo de ayer?
Es el de ayer.
Sí, no. Hasta camina igual de raro.
Oh wow. Beautiful.
He's kind of coming
Yeah.
after me a little bit.
He's defending his territory or her territory. Wow.
So yeah, right. I can cover it if you need it.
Is
But
it
we will try it.
okay?
So we'll try this one. So
What are we gonna try?
collect it.
You try and catch it? All right, with the net or?
Yeah, I think they're
Which
too
kind of
fast
is?
for the
Tal vez sí solo
A net, and then he's gonna
pero si solo
go.
tenemos la red enfrente, él solito se mete.
Can you go in the place with the rock?
Yes.
You can? Okay.
Who's that? That's a big ass lobster?
No, it's a
Or is it
big
shrimp?
shrimp.
All right, you can take the camera if you need it, and get the net ready.
Morning. How are you doing?
I think it's on
Yeah.
the porch. I can porch out if you want to. I'm just not sure I'll be able to porch it.
Wow, look at this ginormous shrimpy over.
Juliana could do it for you. Wow. Look at the size of that shrimp.
Yeah, that's what I was just saying. I was just pointing at this. It looks like a lobster. It's
Oh my
so
God.
big.
He's like 100 octopuses.
Yeah.
That's
What
crazy.
is he eating?
Probably a shark.
Yeah. That,
That's the apex
that's
predator
the
right there.
apex predator on the apex of this
It's
formation.
a sperm shrimp.
He is really big. Hmm.
Not for you.
I know you're cold-hearted.
You feel the
Was that with the octo?
Timothy.
I
It's the
did.
octo, not the
It,
shrimp.
I couldn't help it. Timothy is on the brain.
Tried to trick him to get the shrimp, so it wouldn't take the bait. All right, we'll deal
What?
with that. We'll deal with that after the, not yours, the temp probe.
Oh,
Not yours.
okay. As long as
Sorry,
mine.
You just gave her a hard
Yeah.
time.
And I'm like,
All right,
okay.
so here's what we'll do, okay? When you go, you want to kind of go down over it, so, because if it has a way to escape out the side, it's gonna do that.
Like this?
Yeah, and hopefully it'll try and escape up into the net, and then you twist it,
Okay.
and so it can't get out, and then we'll put it in the bio box, all right? And so we'll try and do that, and it'll all go terribly wrong, and then we'll probably give up. All right. You ready? All right. Start creeping in. Porch is not out, right? Or is out? Porch out. All right, I'll get you really close, okay? Okay. Start kind of getting close there. All right. That's all you, bud.
Whoa.
Can't come down anymore. Getting rock. So tiny. Twist. Okay.
Good
just
job,
let me-
everybody. Good job.
Whew.
Good
job.
job.
Thank you.
Load up, I'll porch out.
So simple, though.
I think so, yeah. Let me,
Simmer. All the animals.
So where's that going to?
porch out.
Yeah, and it's already. Does it fit in one of these?
No.
Yeah, he's gonna have to go where the rock went, I think.
Yeah.
we're
Oh, when the rock.
BB two.
Maybe
To be or not to be.
It is to be apparently. Alright, open them.
Four sevens.
So you might have to come like, see how your arms kind of, the elbows tight? You might have to kind of come more out like right now. Kind of like that, look more like that. That would be a better angle. Does that make sense?
So, but do we put the whole tent inside now?
Yeah, the whole thing goes inside. Yeah, but ideally in the one with the rock, which is the one closer to the group, because I don't think they want to smash that.
The coral is in the other one.
So,
Sorry?
The octocoral.
Yeah, I think the sponge is going to be the one that we really don't want to.
Okay, so as long as it's.
Yeah.
so I can go in
It's-
the coral
yeah.
one? Okay, you can go in the
Okay.
coral one, and if that's easier, it can go in.
Yep.
It can go in here. Let it go and see how he does. There you go. Yeah. You might just kind of have to nudge it a little in there, you know. Go. Alright, you might have to nudge it one more time. I'll see if close no lid does it. Like straight on like that and see if you can scoot the bottom back, you know what I mean? Yeah. Down there we go.
Nice. Well done.
Here.
476.
476.
BB2A. very temperamental.
Yeah, I'm gonna set it down for you. All right, you ready?
All right, ready for the summit.
All right, I just gotta go.
Oh, kind of at the summit, aren't we?
Up and get this temp probe real quick. Okay.
Maybe I can get it from here.
You think so?
Just fine.
Okay. Just let me know if you want me to set it down for you. Is that the whole holder came off?
Oh,
Yeah, the whole entire holder did, huh?
is it just sticking through the?
It's like a magnetic.
Oh, I see.
That's the problem.
I mean, it was kind of cool, but honestly, I didn't totally like that thing.
Probe.
Because it was all in the
Oh,
picture, right?
The
Yeah.
full probe is like,
Oh,
oh,
no, that's not the heat
Ah,
flow
it's not-
probe.
yeah.
Yeah,
not?
that's
okay. Yeah.
no, that's the temperature
Yeah.
probe. Oh boy. Oh god.
Just make sure to yell really loudly directly next to me.
I mean, that's
They will not panic me at all.
Sorry. There's two things that look very similar.
I was on a boat once, it was like 3 in the morning, and on the radio, someone just went, I'll put this on.
Run! No!
We were like, it's
That
it's
is
just
like, how could I do that
is
right
the
now?
direction in there?
Yeah, I'm on a ship also.
Can we put this in the bio box?
On the box.
Is that what you're asking? Or are you asking
No, that box there.
the little Charlie?
Rocking box.
Okay. That reminded me of that.
You said run while you were on a ship? It's kind of a helpless feeling. I'm on a metal island, guys. I can't run anywhere. We are just adjusting something on the porch.
my phone from under the seat of my car.
Yes, it's exactly what it's like. Yeah, exactly. It's like while you're driving, trying to get your phone from the back seat. Yeah, or under the seat, I guess.
You want me to set it on the ground for you? Watch out, those hooks like a foul tight, and that cable's getting real tight.
Maybe like navigate back.
We're doing heat flow probe measurement with our temp probe. Watch out for you.
Oh.
Now,
Unfortunately, to those who are hoping to use Apex Shrimp as your band name, it has already been taken by a company called the Apex Shrimp Company. Yep. I mean, these are important things to share with the public. Oh, did that fall through the grate or the
Yeah.
temp probe?
No, it has a holder that broke off.
Oh,
So you have to hunt the holder in the box.
Oh, God.
And then you have to grab the probe. and back.
okay.
I think just
Doing
all-
some yoga?
It's just always in the way there.
Yeah.
Open for the new sub. I don't know if Bob's will do it, but we have these little rams that we use to do gas packs that fit on the arms.
Oh,
It's super cool to have like a temp probe on it where you just go.
yeah. Yep. Like the heat flow probe.
Yeah, but for the arm.
Yeah.
You can use it to fight apex shrimp.
As exactly.
and rubes.
Oh, cool. What are those?
Few worms.
Yeah, I think so. And clams. Bacterial mats. Ooh.
They're alive too.
Yeah. Cool. It's a little squat lobster. What's that green thing?
Oh, that's one of
What
those
is it?
weird worms.
That is weird looking.
Yeah, we've seen these. Not here, but we've seen these in other places. They're
Huh.
like super long worm things.
That oh, it's moving. It's retracting or
Sanacura.
what is
The
it?
green one?
The green
A
one.
spoon worm.
Spoonworm.
I think so.
Huh. Yeah, I think spoonworm is not taken as far as band names go.
Oh, it's retracted.
Yeah. Cool.
Wow.
Oh, cool.
I mean, oh no, I have no idea.
So it lives in the rock? Or does it have a
Is that all this one?
No, I can do some more of this sub one.
There's always more some.
always more? Oh, cool. What is he doing? All right.
Oh, it's going in there.
Okay. Good night.
Something very creepy.
Wow. This looks a bit like a horror movie. Yeah.
I don't like it. I don't think we should see it.
This is how things go sideways. When you... But look at those giant tube worms coming out of the top of that thing.
Look at all the giants.
Enzoanthids
Why are they here? Nowhere
everywhere.
else? Remember all the
I
sharks
don't-
earlier?
Yeah.
You remember like around Tango Said?
Tango
With the
Said.
top of
Yeah.
It's full of zoanthids.
Yeah.
I don't understand why.
What do zoanthids thrive on? They're just filtering. Yeah.
Grabbing.
Hmm. All right.
More and some more?
Yeah, definitely.
All right, there's some big rocks to my left. You want to check those out?
Sure. Yes, please.
How far is the boat going? They're done moving, right?
Huh. Wow. So we really aren't at the top yet. We're still heading up, right?
We're just about at the top.
We are.
According to my massive RV.
Yeah, the scale's a little off
I know,
there.
I know, it's like 20-meter scale.
Mussels, huh?
Oh, yeah, let's check that out.
It's a good thing we got up here with extra time.
Yeah. Is that a first, having this much time to?
No.
No?
It's a little better than that last summit anyway. Remember that one?
Yeah.
You want to actually park or just?
Yeah, if we could, you want to
Yeah, let's
spend a
take
little
some.
time here? Yeah.
Okay, park.
So mussels are a good sign of active seeps, active methane
And I got
seeps.
a bunch of wacky stuff there, see?
And this whole summit has been covered in zoanthids. And we were just talking, there's another dive we participated in that had a similar composition of zoanthids covering everything. And that was a skate nursery. Skate park.
Yeah, I was on skate park.
Yeah, skate park.
We're just about, we're pretty much there.
Yeah,
Hard
It-
to get much higher.
yeah, cool. Hmm. Mm. Yeah. These are really, this is the only spot where we've seen live mussels, right? And we've seen lots of tube worms and a little crabby.
Yeah. Less than 10 centimeters.
Yeah.
These aren't super big ones,
Yeah, not like the other sites.
No. Some of those look to be like that, you know?
Oh, it's dangerous.
Over there?
Hmm.
The one behind. Yes, sir, that's only with that out.
Joanna, would you like some chocolate?
Hmm.
I said no, and you just.
You said no.
More exploring.
Yeah.
Too late to stop that.
Let's go find more cool stuff.
Looks like something interesting that's on our end.
Yeah.
Go there.
Yes, please. Thank you. More cool stuff.
When are we supposed to come up on one?
2.
Sorry, on deck at 2.
Oh, okay. 1. Yeah.
It's in a box.
On deck at one. Okay, so come up on.
No, on deck at 2, off bottom at 1.
We got time.
Right? Oh, on deck at 1, off bottom at noon.
Either way, we got time.
Yeah, I don't. Time is irrelevant to me right now, so.
I'm gonna turn this way and see what the sonar diggly doos.
So we are at the end of a what? 12-hour dive, including transit.
Started at 1.
Then we'll be back on 1 at 1. 2.
On deck at, oh, we leave at 2, right?
No.
No,
Oh,
on deck
so
at
we're back
2?
at 2, yeah.
Bottom at
More of those bacterial mats.
we are in the, yeah, that's where here, then we will go down to some escarpment after that. So
Huh.
Shells.
A lot of shells here. Oh, more mussels.
The sonar targets here, see them?
Oh, yeah.
Yep.
Oh, black mussels.
Yeah.
Woohoo.
Yeah. You can smell that sulfur.
It looks like different kind of clams.
Mussels?
Yeah,
Well,
like
The
ones
clams
that-
around? Yeah.
the ones are mostly clams.
Yeah.
Yeah.
So cute.
Stop here or
I'm gonna come and put this
keep
in.
on looking?
Yeah. If we could just zoom in a little, see if we get any.
Yeah, but they're
Never see a life
It
Yeah,
looks
we
like there
collected
might be some
but not.
younger mussels, maybe baby mussels.
No, we collected some alive.
That's like the more yellowy.
Yeah,
But
no,
they're
the
pretty
more
much-
yellow, the younger apparently, but you can see like some of them have little, obviously there's snails and things, but also,
That's the same snails we saw last place.
yeah. But what about like this little guy? Can you zoom in? Yeah, is that a baby mussel?
Yep.
Yay, I found baby mussels.
Good
Yeah,
job.
I know. I'm a biologist in training. Yep. I get a star for that. Look at this. There's all over the place little mussels.
I love their colors.
And this little guy. Cool. It's only the past couple sites that we saw the little baby mussels. All right. I only have 8 more minutes, guys.
Oh, look how shiny those shells are.
Yeah, look at that. That is beautiful.
Oh, I see a
Feel bell.
Eel pout. Oh, look at him. Look at the little guy right here. That little, yeah.
Yeah, yesterday
Oh.
one of the samples, Rich,
What?
on one of the samples when we collected the fossilized
Yeah,
whatever, there was a piece of the coral
yeah.
and it was like golden, like it was like mineralization of something
Oh.
and it was like gold.
Wow. Well, if we can zoom in again just to nerd out on that, that's aragonite. So that's a same,
Oh, those shells?
another form of mineral, like calcium carbonate mineral, is that like opal looking, yeah. Out the exterior layer on those shells. And each of these little opal looking, shiny, yeah, they're common, they're often used in like jewelry and things. Cool. All right.
You know?
Yeah, we'll keep going. And then if we do eventually find soft sediment, I mean, obviously not here, but,
Oh,
Wait.
I got a guy.
oh, there's a guy behind you. Look out.
I got a guy. He wants to know about my car's extended warning.
Yeah.
All right, we're just exploring. We got like till noon. I think at noon we come off bottom, so we're kind of heading towards that raggedy looking stuff.
Yeah, I have one till half an hour. It's all good. Big crab on the right. Somebody over.
Big rock on the right.
Big
Crab.
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 Okay. Where?
On the right.
Your other right?
There it is.
Okay, there it is.
Behind the rock.
Oh, whoa! That is a leggy crab. Let's zoom in on that. I mean, true. Good point.
It's just
How is it hide? It's like it's hiding behind that rock. Do you not realize how big you are? Come on.
Like a toddler trying to hide.
Yeah, like putting his hand in front of it, like no one can see me. Right. A big yellow submarine with giant lights on it. Oh, how did I miss you? Oh, there you are. Cool. Is he eating? No. This is my rock. Looks like he's doing push-ups or something. How big is that one? Oh yeah, that's big. Wow. Okay. Yeah. It's beefy. Spiky. Oh. Do we know what kind of crab it is?
Yeah,
Is it? This one? This kind is? Okay. This is it Paralomus?
Brazilian.
King crab.
Looks like it.
Yep.
Deadliest catch.
Yeah, Deadliest Catch. I love that show. That would be a big catch. And as noted, it does appear to be missing a leg. Oh, got into a fight. It's something. Huh. Yeah. Very cool. Yeah, sure. No, this we have bonus time at this point, so we're another benthic ctenophore and a little bit of coral. Yeah, it's a king crab. The scientific name is Paralomus arethusa. What's the CF?
No, it's not this one.
Not that one?
No.
All right. Fail. Try again. It's not in the, yeah, I looked, it's not in here. Okay, it may not be a king crab. We can be sure it's a crab, but that's all we got right now. And it is missing a leg. Yeah.
Yeah, the thing is that king crab is a name for
Many things. Yeah.
It's not a monarchy.
It's not a monarchy? Is that what you said? So many kings at the seafloor. I'll be off the mic soon, don't worry. Oh gosh. Oh, more mussels. Three new patches. Wow, this is the dive that just keeps giving. Look at this, the one in front of, oh, I guess that's a little divot. Wow. It's not an Alaskan king crab. Yeah, maybe check out this bigger pile.
tired or how it's a good time.
So it was actually a deep sea spider crab.
Deep sea spider crab. We have a verdict.
Those are sea spiders. Yeah.
Huh.
Okay.
One, I should do some food aside. I'll send you a picture. Adios, jefa.
Huh. Oh, more babies. Baby mussels. Cool. And sponges and yeah, there's a lot going on in there. Tube worms, bacterial mats, more aragonite. Snails. No eel pouts so far. Cool. Keep exploring. Oh, who's on next? Oh, sorry. Hi, Sydney.
You're gonna try to get
Yeah, we could try getting a heat flow measurement here. Just at least check, see if it's soft enough.
I don't know where it is. We just moved the camera aside. Okay, oh, come on.
You're gonna try and measure on this area?
Yeah.
You're gonna go and scan the room there. See.
Yeah. We'll keep, I mean, we'll obviously keep an eye on the pro when it goes in, make sure it's not hitting anything.
No, it's only just where I see what has.
All right, I'm gonna,
Temperature for this.
we're gonna collect, or try and collect a heat flow measurement here.
I could portion all of it.
First we'll check, make sure it's soft sediment. Obviously there's some rocks around.
I could zoom in.
I'm allergic to air conditioning apparently. I'm going to pass it over to Sydney, who will be with you for the next hour.
So we're just exploring around, so we'll try a deep flow for now. We'll explore the top. And then especially, yeah, towards it. For now, then. Let's see about time.
Are we finished everything?
Sorry? Yeah, we're, yeah, so we're exploring around.
Temperature for. Yeah. Welcome.
Thank you. Take it out of the halfway mark.
Good morning, everybody. My name is Sydney Christopher. Welcome to the Deep Wonders expedition. So, for those of you all joining in now, we are just going to do some heat flow measurements. We've already reached our last waypoint, so we'll just be exploring from there before ascent, basically.
Okay. Could you help me push in the board? I'm coming. Where? Yeah, I'm fortunate.
special place to test?
We're in a good position for the purposes of this primary
Try not to bump into anything. Okay.
Okay, Brendan. Eco probe power is on.
Okay, just give me a couple minutes.
Should I penetrate?
Not yet.
Okay.
No, mine's in my room. I thought it was up there.
Sebastian would have been housed, but this is where it's controlled when it's under the sea. So ROV Sebastian is controlled by a very long, let's say, wire, it's known as like a movable word, that's what they call it. So that's how they connected and get live footage as well as give the ROV instructions so it's going to do. So
Yeah, it was just, I was having a hard time calibrating it.
Who control where the ROV goes, and they also control the very
Okay, I'm recording. You can put the probe in when you're ready.
So just keep an
so
eye
they're
on
able
this
to
is going in.
Yeah.
samples, suction guns, all from this room here. And then to the left of the pilot, we have the dive lead, who is
Full
able to
penetration.
All the way
direct
down?
all of the
Yeah.
activities that happen on the dive. So
Yeah.
whether we collecting video transects, if we collect any samples, anything like that, that is where all of that is controlled from. And then behind the dive lead is the logger. So that's what today. Sydney is our dive lead currently. And the logger basically logs every single thing that we collect, every visual observation species that we see. And of course we have Beatrice, who's our watch lead. So she essentially is the one with all the experience that knows, exactly what we need to do, and she just watches over all of the activities that happen.
Clint,
So yeah.
could I have that bottom right screen here? Just the one with the position of the ROV. Sorry. Just like the one that's on the bottom right. No, of the four split.
That's it?
That one, yeah.
The USBL?
Yeah.
Yeah.
Yeah, that's
Oh, the cruise
not
line,
exact time.
yeah.
It's not that critical.
Good. Good, good.
So you knew about SOI before you came in? Yeah.
There we go.
Alright.
Thanks,
Did you
Clint.
know about SOI before coming in?
How long has it been?
Next month, it's gonna be 2 years.
It's your cousin. I went to undergrad.
My cousin. Oh, BB?
Yeah.
Yeah, that's my wife's cousin. That's
Oh,
my cousin.
okay, okay.
Too much chair.
Yeah, when they're on board, which is past me. Like, what are you doing?
So you still you read?
Yeah, I'm doing my masters now.
So you're doing the masters full time or you're working as well?
Part time, so late hours. So yeah, I'm working part time as well. So it's not a full time job. It's
In a related field or?
environmental science project management. So all good.
Yeah. Lucy. Zach and I will be looking to. Yeah.
Oh, do you go to the players? Like we did last time.
It's warmer. Were you one of them about 3 seconds ago?
No, actually, I put in more press. That's more than gravel. Fairly okay with seasickness. After this, we're going back home.
Sorry. I am on the next cruise, but we're going home. I'm coming back.
Rotation.
Yeah. It's probably going on like 5 in the evening. I'm coming back. Most likely I'll be coming back like midnight. Can't handle the 2 hours traffic anymore. So yeah. Because even if I have to leave home to reach here for 7, I have to leave home at 5 and still have traffic. I was like, the hell with that, I'll just leave home at 11, reach out here by half 11, and that's what I had. I've been working offshore so long, I can't handle rush hour traffic. 2 hours every 2 hours. I'm going home and going to work, sorry. Can't do that.
It's the way of it.
Same place?
Yeah. Could we not put it down the camera?
out, so take a look. There's one problem is matter. You need to get up the rope.
Why can't I play back right then? Just figure out that. Just in case the signal just falls out.
And then if you need to pick it up, probably I'm gonna need to pick it up again. If you need to pick it up, the bottom, okay, first again, and then we Please don't force it.
Yeah.
And porch are afraid to lose on the interface. Forget the word and the interface, so.
Seven minutes left. Yeah, he wants just more.
Okay, everyone, so just bear with us a few more minutes as we're doing the heat flow measurements.
Because they are like
Snake trigger.
Which ones?
The white one.
Couple years. But I mean, it's pretty cold down there. It's very limited resources, so yeah, years. Deep sea corals.
Okay, we're good.
Good?
Yep.
Alright.
So I think we can explore the top a bit more. Places where we haven't been, and then
Going this way.
we will want to come down the hill by the other side. Just explore a bit this,
This side?
like this side here. To go
Okay.
again to this,
11-ish.
like to the edge of the escarpment.
Okay.
So just for a while, see what we found.
Sounds good.
Yeah.
Is that the side you want to come down? You want to come down that side?
Yeah, it's like
Down, yeah.
I think the base of the camera, right? The base of the camera. They put the base on my phone. I think the base of the camera.
Yeah.
Yeah, so it's just we just put it on there.
I agree. There was also this collapse in here. And then on the ridge. Yeah.
Mhm.
Let some more.
Yeah.
Oh yeah. I can show you that. Easiest way. Yeah.
You want to open the portal from the air?
Yeah.
I just think we're gonna knock your
I was just very good. Anyway, I'm stuff as well. So. Yeah, didn't have much more. Oh, then check the days. So what happens now? Sure, up and out.
and then we will come down
You go
here
down
to the edge of the escarpment.
Sure, up and out. Yeah, okay. Drop it down.
Was that an current?
Sorry?
An current?
Lovely.
So another one.
Sunny,
Yeah.
they were asking, can we drop a point? You see, fuck. See our mouse is on the flater mouse there,
Yeah.
over here. We just drop a point in over here on it. If we zoom out, we should be able to get into it. No, you won't see it on there, you'll see it.
Yeah, for
Hold on, you do it.
See if we can figure out where we are. where the mouse is. Over there.
What direction is the top of the screen there?
Just hold on and I'll get you.
The top to the north.
So that's north now, right? North up.
Okay.
That's north up.
Okay, and show me the RV.
There's the ROV, is that little red yellow? There's the ROV.
We need to go north.
You know what, if you went
So, right.
straight line to 11,
So I think
Make it bigger.
no, you want me to go between 11 and 12, right? To the east.
do you want to, yeah, there you go, you have a chat there.
So yeah, if I just go straight
If
on
you
this
go
line.
straight from
Yeah,
that line to
exactly.
11, I'd say it would cut across there,
Yeah.
down that valley.
Here. Just not again to 11, just to this edge.
Or you want to go down there?
Yeah. So I think if that's not there, I'm facing in the direction here
Yeah,
anyway. So
I think you're good.
somewhere like
that
here, you know.
direction, yeah. Because
Yeah.
we will come up like here, I think.
Okay. So you want between 11 and 12, somewhere around here, right?
Yeah.
Okay.
I'm going to come out of this north upper,
Yeah.
so I can actually
Fish.
see what's going on.
Want to bring it so down now. You're gonna come on this way here.
Right where my mouth is, like, so yeah, I'm just gonna be coming southeast.
Okay, everybody, welcome back. So we just finished doing a heat flow
Where are you, Gerald?
measurements, and we're just gonna do a little bit of
We are
exploring.
heading
before
south,
we
southeast,
maybe
but we're not
take
sure
out
exactly
sounds.
the path we're gonna follow.
So I don't know how many of y'all know and
on the
have been
area,
out for the entire
if you can
dive,
follow
but
us.
you know, we had a pretty early start.
So far.
We actually had about 12 waypoints that we actually covered this morning. I think we started somewhere around 4 a.m. Yeah, a.m. Science doesn't sleep. Yeah, so just finishing up. Feel free to take a little rewind, check out all the cool things we've seen so far. Sponges, crabs. Bigger crab.
Just asking. Move a little
More
faster.
fishes.
Where are you, Gerald? Me again, sorry. Can you start a little bit
That is
faster to
a
catch up with us?
Yeah,
sure. I'm making that 26 flying
So some of the organisms down here actually use the corals as like mounts. Climb up and kind of grab things from the water column from up there.
Yeah. Yeah.
Actually does pretty well to kind of hide them behind structures as well. So really interesting.
Assuming this coral for a bit.
Mm.
This one either. I don't think we have this one either.
We don't.
No.
But the other one was bushare. This one's bushare. The other one's very sort of the coral and like.
Let's take a piece of this.
Please?
Take a piece.
Okay. Can we
See?
get a
Sure.
small piece of this one, please?
Yeah.
You want a sample?
Yeah.
Bridge, Rovi.
Okay, so just pausing for a second here.
We're gonna stop
a
here and
sample.
sample. You can come towards us just to get closer, about 50 meters
Really
closer.
pretty coral here. Just take a closer look. Sea spider to the side there. For those of y'all who were tuned in, I think it was yesterday, we saw 2 of those towards the end of the dive as we were doing the descent down a slope at the end.
First.
Yes, please. Can you zoom in a bit when it goes up? Thanks. You zoom in closer to this area. Some pictures? Yeah. Okay, let's go ahead and sample. I'm thinking maybe the section on the left.
What section is it? That's on the left.
On the left.
In a coral river.
Okay.
Maybe a piece.
Okay, so
Ready? Yeah, man. So you might just have to move a little closer. Okay.
So for this one,
Where is
since we
it?
want two snips, we should sample one and keep the other one to check.
okay, do we want like a bigger one and a smaller one, or
It's like maybe here and then maybe down there,
maybe?
something like that.
Yeah.
Like,
Okay, so this one and this one.
yes.
Okay.
Oh, yeah, let me go a little closer. Let me go a little closer here.
Did you log the sea spider? Leave over that now. Very good. Where is this gonna go? Where do Okay,
you
so
want it?
coral quiver 2.
Well, route 2.
Yeah, so we'll take
Yeah.
actually 2 snips. The bigger branch and then the smaller one. The ones right next to each other.
So there's 2 on the left. Right?
Yeah.
Those two.
Yeah. Yeah.
Good size?
Yeah, correct river.
Okay. Yeah, that's good.
You like
just
You're gonna get the glove also?
it might it's a little
That thing.
Might fall.
yeah. So just try to get it in the
Take it off.
yeah, open.
What sample number is this?
Still a piece in it, just
477?
shake up.
Okay, great.
There you go. Lovely. Is that enough or you want more?
Yeah, I think that's.
Do you want more or is that
No,
enough?
that's good.
Okay.
Close.
Yeah, close.
Yeah. It's not on. Close. No.
Okay, great. Thank you. Let Aaron do like more pictures since we're going to get on shot. Okay. Let's continue on. Okay, so for those of y'all tuning in, we just collected 2 pieces of pink coral, 2 little pieces. We're gonna have one for our museum collection at the UE Zoology Museum. Please come by and check us out. Lots of interesting specimens so far. And the other one I believe we'll be using for cryo. Okay, so we are going that way.
Come back out or just head south?
South. Yeah, that way. So, I think we're going down this.
Yeah, I think
around,
so.
so
Going south
yeah.
is
So come this way.
Yeah.
Okay. Come it down. Right. Right. Yeah. South. Right. No, no, just heading south towards. Just kind of, yeah, just past 9.
Yeah, what we are trying to do is explore this edge here.
Yeah.
Explore it. So, yeah, down there where you are, south.
This edge here.
Yeah, I mean, if you go south from here, you
So where exactly you want to go? Like here?
yeah, just like
This
the
part
edge
here?
of the blue part.
Parallel with the other.
Pocket?
Parallel with the
Yeah.
back of
Lower.
it. Like parallel to the
Yeah? No, no, further down.
like here. Yeah, there's you see the edge between the blue and the green there.
I think.
Okay, onwards.
Very interesting waypoint.
Update waypoint.
You want to pull in the coral thing just so we know?
Cover them? Do I need to?
Pull in the coral cover just so we don't
Oh
have.
no, we just need to get off the sea.
Yeah. Nice, nice. Sea star. as we are approaching the top of the hour, just out of curiosity, y'all can name a couple of things y'all are interested in seeing. You know, we have basically 1 week left of the expedition. Still a chance we might see some of the stuff you'll have been expecting.
What's the point of going down there?
I already see it, so.
They won't see anything, they'll just get forward.
So we'll switch that then?
No, I'm just coming down sideways. I was just moving forward a little bit just to get out of that way.
Okay. Okay, yeah, continue on.
Or seepage, I'm just slowing down.
Yeah, roger.
Okay,
So
so
at that point you want to get to quicker, or you just want to look around?
Look around. Yeah.
okay, so it is 12 o'clock, I think, local time. So that brings me to the end of my shift. I will pass you guys on to the amazing Bia.
Hi again, man. Hello everyone, I'm Beatriz Naranjo from Costa Rica, independent consultant collaborating with the University of Costa Rica and Species, and we are exploring down to 1,000 meters, a bit more, the exclusive economic zone of Trinidad and Tobago. Buenos días, buenas tardes. Mi nombre es Beatriz Naranjo, bióloga marina de Costa Rica. Soy consultora independiente, colaborando con la Universidad de Costa Rica y diversas organizaciones como Species, que es una de las principales organizadoras de esta expedición en aguas de Trinidad y Tobago. Ahorita estamos explorando la cima de un pequeño montículo que mapeamos anoche y donde encontramos algunas señales de emanaciones, que son estos sitios que hemos estado observando a lo largo de la expedición, llenos de almejas, de mejillones. Hoy los encontramos principalmente en la cima. Ahorita estamos descendiendo a la cima y explorando los alrededores. Parte del objetivo es entender hasta qué punto estas emanaciones tienen influencia en las comunidades biológicas, y parte de nuestro trabajo es muestrear, tomar muestras para genética, para describir especies en caso que sean nuevas, tener nuevos registros. Hemos observado muchísimas especies, que eso es la primera vez que se registran para el país, y algunas para la región, e incluso algunas especies que creemos son nuevas para la ciencia. Principalmente emanaciones de metano es lo que encontramos. A veces vemos burbujas, pero aunque a veces no hayan burbujas de estos gases, podemos ver actividad bacteriana, que es señal de esas filtraciones. Unos corales negros, erizos, corales hongo. Y una de las razones por las que ahorita no vamos tan cerca del fondo es porque vamos cuesta abajo, y si nos acercamos mucho al terreno, la parte trasera del robot choca con el fondo, y son esos momentos donde ven que se levanta mucho sedimento. Así que estamos intentando bajar esta colina y encontrar un terreno más fácil de explorar. Is it steep?
Oh no, I can turn around and
Okay.
go straight with that. Y bueno, el buceo de hoy está en la 6ª región que hemos estado explorando, y nos concentramos en seguir una línea de puntos que observamos en el mapa como puntos de posible burbujeo. Esas son mediciones que se hacen desde el barco cuando no estamos utilizando el robot.
y utilizamos una sonda multibeam que emite señales acústicas que rebotan con el fondo, y cuando rebotan con posibles burbujas podemos ver eso en el mapa, y así sabemos que puede haber algo interesante pasando, y es parte de la información que usamos para decidir en qué sitios vamos a bucear. Así que lo que hacemos es intentar seguir esos puntos que ese mapa nos sugiere que hay burbujas, y intentamos buscarlas. No siempre las encontramos, pero lo que hacemos es explorar alrededor y intentar cubrir ese margen de error que los mapas pueden tener entre sí. Muchos de los mapas que estamos usando, o casi que todos, son mapas que hicimos durante la expedición, ya que son zonas poco exploradas. Antes de esta expedición únicamente existieron 2 expediciones previas, en los 80s y 2014. Y una de las grandes diferencias de esta oportunidad es que tenemos muchas personas locales, 15 de 25 científicos, científicas a bordo, son personas de Trinidad y Tobago. Those clamshells on the left, right? Estos sitios de emanación no son permanentes, pueden apagarse con el tiempo, y lo que hacemos es buscar evidencias como esta que estamos viendo en este momento, donde tenemos conchas, actividad bacteriana, y algo muy interesante es que, a diferencia de las aguas someras, donde la fotosíntesis por la energía solar tiene gran importancia en las cadenas de alimento, a estas profundidades una de las principales fuentes es en realidad la quimiosíntesis. Y entonces cuando encontramos mejillones, almejas, incluso esponjas, camarones, toda una serie de organismos tienen relaciones simbióticas con estas bacterias. Y estas bacterias, lo interesante es que el metano y sulfuro, que son los principales gases que salen en estos lugares, suelen ser tóxicos para los seres vivos, pero estos animales, gracias a estas adaptaciones y esas relaciones mutualistas que establecen con las bacterias, son capaces de alimentarse. Estos, por ejemplo, estos puntos o estos agujeros que ven ahí blancos, esos son actividades bacterianas. So I've been explaining about the objective of this dive. We've been exploring a set of waypoints that we established last night thanks to our multi-beam mapping. It was suggested that we would find some seepage areas down here, so we've been following these dots, trying to see evidence of methane, and what we are trying to look is we try to see all these communities that include mussels, clams, shrimps that are really adapted to this kind of environments where gases like methane and sulfur are usually toxic for life, but all these animals have adaptations and have established relationships with bacteria that are able to evolve or develop this methane into ways that are actually ways of feeding. And right now we are going downhill So that is one of the reasons why we are not super close to the bottom, because when the back part of the ROV hits the ground, you see all this sediment. So we are going downhill trying to find a plain terrain.
You're like, I think you're in this crevice here, where my mouse is. I think you're like here-ish.
Oh,
I thought I was still
so
coming
was
down
the
the
ROV
hill.
in that map?
Yeah,
Ah, okay.
but I'm coming up the hill now. No, I'm not that far down. I'm level with waypoint of level. Actually, no, maybe you're right.
Yeah.
Yeah, past the level. Yeah.
Oh, really?
Yeah, I think you're coming down this way.
Oh, cool. That's what I wanted to see. So one of our biggest findings today was a carnivorous sponge. We have three experts, sponge experts on board, and it was at least 40 centimeter, I would say. That looks like how do you call them? Polypop sponge. Can we zoom in on this?
Yeah.
I think it's a coral, but might be another sponge. I don't know. That's what I saw here earlier. I was like, this was here, like, no, what's the sponge? So how do you call the carnivorous sponge?
Death ball sponge.
Death? Death.
Death ball.
Wow.
This
Okay.
one in front or the top one?
No, the white one in front. Yeah. So the common name for the sponge we saw today, and it's dead ball sponge. So quite an interesting name because they trap organism. We saw an amphipod, a type of tiny crustacean, some sort of a shrimp, and we believe the sponge was eating this animal. Yes. Estaba explicando que uno de los descubrimientos que hicimos hoy fue una esponja carnívora de unos 40 centímetros que literalmente atrapan a otros organismos. Beautiful. This is some sort of sea pen, coral. You might want to collect this, but I want to see the polyps a bit more. tal? ¿Todo bien? I think so, but I want to see the polyps better. We have here our artist at sea, Marco Barotti. Barotti, right? So
Hi everyone. I hope you're having a good day.
So Schmidt Ocean has this artist at sea program, and Marco is here looking for inspiration, but I think he's too inspired now.
Yeah, I think I found a lot of it.
So hopefully this expedition will help him to keep creating his amazing art.
And it will go to Venice in 2027.
Venice 2027. Oh, cool. So we're trying to get a closer look at this white animal in front of us. The one on the left is a black coral for sure, but the one on the right, or the white one, we are a bit confused or about what it can be. I do see 8 tentacles. You count 8? And since the stalk is black, at some point we thought it might be some zoanthids living on a dead stalk. No, we are focusing these ones. Can we focus these ones? Yeah. So yeah, I do think it's a octocoral.
Focus on the front. So you top point there with light. Press it to make on and off.
Yeah, I will take a snip.
It's gonna focus in a second.
Beautiful.
So sexy.
Okay, so we will try to grab a snip. This guy.
So maybe
I think it can go in a coral quiver.
from here.
Yeah. Yeah, hopefully the little guy stays as well.
Cool.
The little guy. The little guy.
I'm
El coral anaranjado es un coral negro que hemos estado viendo a lo largo de la expedición, pero el blanco nos tenía un poco confundidas. Así que vamos a tomar una muestra para observarlo mejor por acá. Una vez tenemos todas estas muestras, cada muestra recibe un código único y tomamos diferentes submuestras para análisis genético. Mantenemos otro fragmento para mantenerlo como un voucher o como un testigo en colecciones biológicas con la que los taxónomos pueden llegar a una mejor decisión al analizar estas muestras. Sin estos testigos es imposible describir nuevas especies, así que intentamos tener el mínimo impacto posible cuando hacemos todo esto nuestro trabajo, pero el valor de lo que hacemos, para lo que hacemos, estas muestras tienen un valor muy significativo. Cuando es posible intentamos tomar un fragmento para que el animal pueda seguir creciendo. I think we can grab the whole thing. I think it might be like a kind of, yeah. Yeah, well, we will bring everything. Yeah. Yes, better. Yeah.
Orange.
Oh, really? Both are attached? Oh. You think so?
It all came up with that.
What do you think?
It all came. There's no way we could fit the black.
The orange is gonna.
I'm not sure if we should abort.
Where you gonna put that?
Yeah.
Me too, it's full.
Yeah, no.
We'll try and get it from the bottom.
Okay, try to cut it.
Yeah.
But if the thing will come up, I prefer to
Leave.
abort. Yeah.
Okay.
I don't want the orange one. Yeah, great. Thank you. Entonces, en este caso intentamos tomar un fragmento, pero vimos que el tallo es muy resistente, así que preferimos tomar toda la muestra, porque lo que iba a pasar era que también íbamos a recolectar el anaranjado. So I'm not sure about this organism, but I do think it can be a kind of soft coral or octocoral living on the stalk of a black coral. But yeah, it's a really interesting sample. So there are some kinds of octocorals that do find surfaces to grow. And in my opinion, this is one of those cases. Sometimes they are attached to rocks, like the Alcyonidae, for example, family. So there's actually a group of octocorals that is called Stolonifera, and it's an example. I'm not sure if this is one of those cases, but the name refers to the way of growing or spreading using stolons. So like horizontal or let's say creeping stems. So it might be one of those cases, but we are pretty sure that the stock is from a black coral. We actually have a blade on the robotic arm for those cases, but sometimes that's not enough to cut them. That's how strong they can be. This one here is another example of a black coral. The one close to the lasers right now. It's a really diverse group, and especially on the deep sea. Just in a few places in the world, they can actually grow in shallow places, depending on the region. But for sure, it's a group that has developed a lot of adaptations and success in the deep ocean. Also deeper than 1,000. So we are close to the end of our dive today. Tomorrow we will dive again, or well, later today. And it will be our last dive deeper than 1,000 meters. After that, we will be exploring shallower areas, including mesophotic sun. That means places where the light of the sun still reaches, but the ones that start to be darker, let's say. So we will be exploring the mesophotic at the end of the cruise, we have 1 week more ahead. So I invite you to keep following the expeditions, and the next dives will be quite different from the ones we've seen so far. It's a sea cucumber, completely covered in dead shells, and the ones that look like teeth, like shark teeth, are actually planktonic mollusks that die and then they cover the seafloor with their shells. Este buceo que está llegando pronto a su final y el que vamos a hacer hoy más tarde o mañana, en nuestro plan es enviar el robot de nuevo a eso de las 11:30 o medianoche más tarde. Van a ser nuestros últimos buceos por debajo de los 1000 metros y es otra de las esponjas carnívoras que encontramos hoy. Entonces, a partir de pasado mañana vamos a estar explorando zonas más someras. Vamos a estar explorando zonas más someras para finalizar nuestra expedición. Nos queda una semana o un poco menos de buceos, así que para quienes nos han estado siguiendo, los buceos que vamos a tener en los próximos días van a ser bastante diferentes. Vamos a explorar también la zona mesofótica, donde la luz del sol aún llega, pero con menor cantidad, pero son ambientes muy, muy distintos al mar profundo como tal. So this is one of the death ball sponges or carnivorous sponge that we collected earlier. Can you zoom in a bit more, please? I just want to see if there's something attached, like amphipods, or we saw some amphipods in the other one. And we think it was part of the meal from this sponge. So we're trying to focus and see if we can find possible praise of this deadly sponge. Wow. Una esponja carnívora. Hoy hemos visto varias en la expedición, pero ninguna como esta. Hoy ha sido el día para esta esponja. Y creemos que estamos viendo diferentes estados de estos tentáculos. No sé bien cómo se llama en el mundo de las esponjas, pero ha sido muy interesante. Hemos visto algunos organismos atrapados, posibles relaciones simbióticas con gusanos que intentan buscar refugio también en estas esponjas. There's also something here. This one. I know it's part of the sponge. worm. Cool. The worm. Can you leave it with the worm for a bit without moving the camera just to get good video? Yes. Okay. Yeah. Well, let's get some more video from the base, maybe. You can turn them off, maybe. Just after this. And well, we actually had a really good sponge lesson earlier while we collected the first carnivorous sponge. I will not try to repeat it because I'm not a sponge expert, but I invite you to go back in the video and hear Dr. Shirley Pomponi, who explain us how these carnivorous sponges were first discovered. What is this thing here?
Yeah, the previous sponge was right towards the beginning of part 2.
Yeah.
So if you're on YouTube, scroll on back on part 2. We were doing a little BBC project work right at the start with the sponge, but then after we finished up with that, we collected it, and Shirley gave a great little talk about it.
Entonces estábamos explicando que la primera esponja carnívora que recolectamos hoy, que está al inicio de la segunda parte de este buceo en el YouTube, en ese momento la doctora Shirley Pomponi, que es una especialista en esponjas, nos dio una clase magistral sobre esponjas carnívoras, cómo fueron descubiertas, y nos dio muchos datos curiosos, así que les invito a buscar ese momento del video. También estuvimos haciendo algunas grabaciones de la BBC, así que si ven que la cámara no está enfocando la esponja es porque en algunos segmentos cambiamos la cámara para tomar video para futuros documentales, así que pueden ir antes de eso, y en ese momento es donde la doctora Shirley Pomponi nos da una excelente explicación muy interesante sobre estos increíbles organismos que atrapan su propia comida. Así que we want all of it. They went to the base the other time. Were you here? Yeah. I think it's the only place where you can go because it's quite big.
So
it's another one of these.
So we can go in the same one?
Yes. Creemos que esta podría ser una nueva especie, así que es la razón por la que estamos recolectando nuevamente la esponja. Usualmente cuando se describen especies se necesita un holotipo, que es el organismo principal con el que se hace toda la descripción, y se recolectan también paratipos, que son los organismos que acompañan a ese organismo o animal principal, donde se describen todas las características que pueda tener. Yes. It's so cool, the roots. Yeah. So yeah, let's get some zooms for the thing and also the roots. I think the other one didn't have this roots so intact. The other one. I did? Okay. I was too like...
I think they got a lot more mud on that one.
Yeah. Así que pueden ver la raíz de la esponja, muy interesante. Las esponjas son organismos filtradores y se alimentan de los materia orgánica y que pueda estar en esa agua, pero las esponjas carnívoras tienen definitivamente un modo de vida muy muy diferente y este es un hallazgo increíble. No es muy común encontrarlas. En la expedición hemos encontrado otras esponjas carnívoras muy muy pequeñas. Pero en mi opinión ninguna tan hermosa como esta. Are you happy with the video?
Yeah, thank you.
Happy. So we also have Megan Coughlin here, another of the sponge experts of the expedition. Should we get a Niskin also? I think I forgot about the Niskin. We can. Entonces para esta esponja haremos toda una descripción morfológica, un estudio genético y también tenemos un investigador a bordo que estudia el ADN ambiental. Eso significa ADN que se encuentra flotando cuando un organismo, por ejemplo, cambia de escamas, pierde algunas escamas, como en el caso de los peces, o en materia fecal, o en el mucus que lo cubre, etc. Entonces, parte de todo eso queda en el agua, y cuando se filtra esa agua y se hacen estudios genéticos, podemos tener una idea de todo lo que existe, aunque no lo veamos. Es una técnica que ya lleva algunos años, pero que sigue siendo una técnica muy novedosa y cada vez se van mejorando las técnicas para estudiar el ADN que se encuentra flotando. So as you can see in the bio box, in the bottom we have the one that we collected earlier. The reason why we are collecting both is because this has the potential to be a new species for science, and when you do species description, you have what is called an holotype, which is like the main animal that is, or organism in general, that is used to make the main description. But you also need other animals that will help with the description. So these are called in the taxonomic word paratypes. So this second one can be used for that, and it also helps to compare and to actually have a better idea of the variation that you can find in the species. So we're trying to accommodate this large sponge in the bio box. It's the biggest one that we have. So the pilots are doing their best to not break it and being able to bring the whole sample intact to the surface. And luckily we also have all these tiny animals that we are seeing on the sponge that might be or possible prey that the sponge is eating, or some associates that are taking advantage of the sponge. Thank you, that was awesome. Good job. Okay, so after this, I think we are ready to come up. It was amazing to finish this dive with another carnivorous sponge. Oh yeah, sorry. We need a Niskin, yeah. Thank you, Megan. I almost forgot about the Niskin. So yeah, we have some bottles that we've seen these numbers here. These numbers are connected to what we call Niskin bottles. And these bottles close once the pilots hit each number. So with that we are able to take water samples that will be used to filtering and try to study the environmental DNA and relate this analysis with the sponge. What number is that? Yeah, the sponge 480. Así que esto que acá, que ven aquí, estos números son números asociados a botellas Niskin que recolectan agua. Y es otra de las investigaciones que hacemos. Y con esto vamos a finalizar nuestro buceo. Les invitamos a seguirnos en unas horas. En unas 12 horas tenemos presupuestado que vamos a estar iniciando la nueva inmersión, que va a ser nuestra última inmersión por debajo de los 1000 metros, como les explicaba anteriormente. Así que espero que nos sigan. Gracias por escucharnos. Thank you all for listening. With this amazing sponge sample of water, we will finish our dives here. But remember that the water column is always full of amazing, incredible animals. So all that is also broadcasted. So thank you and see you in at least 12 hours.